So bioinformatics is going to be the representation, storage, retrieval, and analysis of biological data. And with biological data, we mean sequence, structure, function, and interaction. So these are the main categories of biological data that we are going to be interested in. Okay. I had a really nice definition from one of my mentors. He defined bioinformatics as transfer of information from known to the unknown based on similarity. Okay, so we have a gene that we've studied for many years. We know a lot about it. We have other genes that we don't know much about. To identify the function, the biological role of these unknown genes, we see how similar they are to genes that we've studied and try to sort of get some deductions based on that similarity. We transfer the information from the known genes to the unknown genes that we have not characterized yet. Bioinformatics is a hypothesis-generating field. It's a hypothesis-generating study. So you look at your data, you come up with some suggestions for further experiments that can either confirm or refute what you think is going on in terms of the underlying molecular mechanism of these biological functions. Bioinformatics is also about discovery of patterns in the data. So if you have a bunch of sequences that have the same role, or say they all go to the ribosome, they all go to the mitochondria, you may ask, is there something in common between these genes that cause them to be targeted to mitochondria? To find that, we would analyze the sequences of these genes, the DNA sequences, the sequence of characters, and see if we can find a small pattern, say ATA, maybe that's giving them the targeting effect to mitochondria, right? So looking at a sequence of genes, trying to find a common core is going to be the problem of discovery of patterns in the data. All right. Let's look at some of the questions that we can address using bioinformatics. So these are just some samples that illustrate how bioinformatics is used nowadays. The sequences of many thousands of genes are now available. So that means for every character in the genome, we know their sequence. So the human genome is 3 billion base pairs, and we have not every single letter in the correct sequence. There are some portions of the human genome that are difficult to sequence. Even though we say the human genome is sequenced, there's a lot of sort of question marks in different portions of the genome that we have not really fully sequenced yet. But we'll just assume that we have. There are some reference genomes that are widely accepted to be the sort of the ground truth genetic sequences of humans, and we have that type of information also for some of the other species. And these sequences are available freely online for anybody to go and analyze. So you can download the entire human genome, you can download the entire chimpanzee genome and see how similar they are, where they differ, and so on. Okay. So looking at a number of sequences, we can ask what do these species have in common in terms of their genetic sequence. And a figure like this shows species as arcs on a circle, and the connections that you see going within the circle mark highly similar regions of these species. Okay. So visualization of the data is as important as the problem that you are trying to address. Sometimes the result that you get is just this visualization itself. Okay. In addition to the visualization, you also want to come up with some statistical measures to sort of answer the question that you are addressing, such as are humans and chimpanzees closer to each other than humans and chickens? And you have to quantify this similarity. How there are going to be many different characters, but you want to find out what percentage of the sequences are similar. Okay? And we're going to see methods for quantifying the similarity between two sequences. So if you take any, say, two words in the dictionary, say calculator and computer, how similar are those two words. There's going to be a process of aligning them, meaning you take each letter, one letter from one word, another letter from the other word, and put them on top of each other. So C with a C would make sense, right? Calculator and computer. Do you then align A with an O? You don't know. So it may make sense to align, or it may make sense to align it with something else. So the alignment will then dictate the similarity between those two words. If you align them, then you can count the number of times that you have the exact letter from the two words, and then the percentage of the exact letters will be their similarity. That's one similarity measure. Okay. In the next lecture, we'll see how this process is done, sort of in a more systematic way, and how we can score different similarity measures between two sequences. Once we look at a species and its genome, we would like to know what genes it has. And that, again, can be done based on similarity. If we've studied humans very well, finding a gene in chimpanzee that is similar to a human gene, we can then identify this chimpanzee gene in a way, right? So if they are similar, we would then assume that the chimpanzee gene is doing the same thing. It has the same biological role, all right? And this question can also be addressed using characteristics of the genes. Looking at a long sequence of DNA, parts of the DNA that are genes will have certain characteristics, as we will see later in this lecture. They will have to start with a specific starting site. They will have to end with a specific ending site, so that the enzymes in the cell can detect those initiation sites and start transcribing that gene. Okay? There are a lot of signals within that sequence of 4 letters that the enzyme will come and detect and start doing their own thing. We can make use of these signals to process the DNA as if we are sort of the enzyme itself. So looking at the characteristics of these enzymes, we would then be able to identify which portions of the genome form genes. Okay. All right. Another question is what a gene does. So given a gene, how can we identify its functionality? There's going to be many types of information that we can use. Similarity is the big one, but we could also make use of, say, amino acid composition, right? So if it's a gene that has a lot of hydrophobic amino acids, then that may say something about the environment that that protein will be in once it's produced. Okay? It may be targeted into the membrane if it's very hydrophobic. Okay. If it is similar to a gene in a different organism, we might be able to make use of that information to determine what it does. If it's a certain enzymatic site, it may be driving a certain biochemical reaction. Okay. All right. So a lot of these questions can be addressed using similarity or using the information that we have extracted from known genes in the past. And all of that information, most of it is available online in public databases. For the expression topic, one question that we would be able to address is which genes are responsible for prostate cancer. And a typical experiment that the researchers would do to address this question is take prostate cancer cells and take control cells, healthy volunteer cells, and compare the two in terms of their gene expression levels. Okay, so a heat map like this shows expression of many different genes from many different samples. So the columns are samples, the rows are genes. A red means it's highly expressed. This gene right here there are many copies of it being expressed in that first column, which is why it's red. And we can then see if there are certain genes that separate prostate cancer from the control gene. Say that gene is highly expressed in prostate cancer cells, but not expressed in control cells, then we can deduce something from that and say this gene is important in the prostate cancer. Either it may be causing prostate cancer, or it may be sort of a marker for prostate cancer that we can make use of. Okay. A lot of these studies are correlational. In order to find whether there's a causal relationship between our findings and the and prostate cancer, we would need to do further biological experiments. For the network topic, a question that we can address is how can the biofuel production be optimized? This figure that you cannot really see on the projector is the network of interactions and reactions in a cell. A small portion of it is produces this molecule, 2-methylbutanol, and if our goal from, say, an industrial perspective is to increase the production of this molecule, we would then be able to ask which other reaction should I change, should I modify, so that the production of this product goes up the most. Okay, and this can be solved using differential equations. If you have the reaction kinetics, you can make simulations and see if changing one of these arrows in terms of their reaction speed will improve your production. Okay. Any questions about different topics and different questions that we can address?